G protein link receptors are plasma membrane receptors that function with the help of a G protein. These receptors are extremely widespread and diverse in their functions, such as embryonic development and sensory reception. All G protein link receptor proteins have 7 alpha helices spanning the membrane.
GDP is bound to the G protein. The protein is inactive. The receptor is activated when the specific signal molecule binds to it, changing the receptor's shape and binding an inactive G protein, causing a GTP to displace a GDP. The activated G protein dissociates from the receptor and binds to an enzyme, therefore activating it, causing a cellular response. The G protein also functions as a GTPase enzyme and hydrolyzes its GTP to GDP, returning the G protein to its original state.
Ligand-gated ion channel receptors are membrane receptors that act as a gate when the receptor alters its shape. Once a signal molecule is binded to a receptor protein as a ligand, the gate either opens or closes, which allows or blocks certain ions such as sodium or calcium through the receptor channel. Ligand-gated ion channel receptors remain closed until a ligand binds to it. Once the ligand binds to a receptor, the gate is opened, allowing specific ions to flow into the cell, which produces a cellular response. After the ligand dissociates from the receptor, the gate closes, blocking further ions from entering the cell.
Receptor tyrosine kinases are membrane receptors that are able to trigger multiple signal transduction pathways at once, aiding the cell in regulating and coordinating several aspects of cell growth and reproduction. The portion of the protein extending into the cytoplasm functions as an enzyme, tyrosine kinase, that catalyzes the transfer of phosphate group from ATP to tyrosine on a substrate protein. The single receptor tyrosine kinase is able to activate 10 or more transduction pathways and cellular responses. Receptor tyrosine kinase proteins exist as individual polypeptides before signal molecule binding. Dimerization results in a dimer that activates the tyrosine kinase region of each polypeptide. Each tyrosine kinase adds a phosphate from a molecule of ATP to a tyrosine on the other polypeptide. The activated receptor protein is recognized by inactive relay proteins that bind to a specific phosphorylated tyrosine, changing the relay protein's shape and activating them. Each activated relay protein triggers a signal transduction pathway that produces cellular responses.